Alright, it's working. Hey guys, so my presentation is on predicting child health with gut microbe development trajectories. And that's the article's name, and my name is Saket Singh, and today is February 15th. Um, so yeah, let's get started. On the top right, you have pictures of the gut, and in the bottom right, you have an image of what the microbiome in the gut could look like. All right. I kept the PowerPoints kind of brief, but because previously it was really long, but I'll elaborate on each point. So, the study focus is to investigate how early life microbiome profiles predict the long-term health outcomes, and it focuses on developmental trajectories from infancy. So, this whole article was about pediatrics and children and the gut microbiome from infancy. And the whole healthcare shift is to advocate from shifting from pediatric healthcare strategies to more preventative care. Like, for example, cardiologists and stuff, they focus more on preventative care when other healthcare providers, like surgeons and stuff, they are more focused on reactive treatments. And the expected outcome is to have potential for the early identification of risk factors for different chronic conditions. These conditions can be they can range from asthma and just like immune system infunctionality to severely chronic conditions. And by fostering this potential of early identification, we can enable well-timed interventions, which is what this statement said. The significance of this study is to highlight the role of microbiome diversity in early childhood and its long-term impact on health. And it basically just like the statement says, it's a shift in pediatric health management. So it basically just paves the way for a paradigm shift in pediatric health management. There we go. Yeah, I just skipped a slide. Alright, so the research question we'll be discussing is how early life gut microbiome profiles and predicts long-term health outcomes in children. So, how do early life gut microbiome profiles predict long-term health outcomes in children? And the goals of this study was to identify distinct microbial community types, and in the study that's referred to CTs, which you'll see later, and identify them in early childhood and their development trajectories, and link these trajectories with health outcomes at age 5, and that includes allergies, asthma, sometimes obesity, like BMI percentiles. The research impact of this study is to enhance and understand the microbiome's role in children's health and guide intervention strategies. And we want to guide these strategies because it's a big shift in pediatric healthcare, which was it's 50-50 in terms of interventional strategies and like specifically preventative versus reactive healthcare. But having more of an emphasis on preventative healthcare when you're a child, in my opinion, is a lot more healthier because you have a lot to go. Like, you have a lot to grow. You should not be reacting to a condition when you're a child. So, yeah, that's the impact of the research. And then some background. The gut microbiome is essential for digestion, immune regulation, and overall health. Its development in the first 3 years is crucial for long-term health outcomes, which you'll see later. And the context of the research is that previous research has focused on small-scale or cross-sectional studies, and it limits the understanding of microbial development. And the purpose of this study is to study it longitudinally and not limit our understanding. And some recent advances are advancements in 16S RNA, which is a whole gene sequencing thing that I'll discuss later, and metagenomic shotgun sequencing, and that has also been an advancement recently. And all in all, it just enhances our ability to understand the complex interactions within the gut microbiome and use them to process data and get certain data that we can, like use to foster this whole preventative healthcare atmosphere that I was talking about. But obviously, we still struggle with some knowledge gaps. Technology can always be better. Despite these advances, there's still a gap linking specific microbiome trajectories from infancy to specific health outcomes in later childhood. And these specific microbiome trajectories, again, affect things like asthma, obesity, irregular immune function, which is the specific health outcomes it is talking about in later childhood. And so the overview of the methodology, I have a couple slides of methodology and they're named different things, but this is just like the whole overview. So to introduce the techniques, it utilized 16S RNA, which I discussed, gene sequencing, and metagenomic shotgun sequencing to analyze the gut microbiome. And these techniques as a whole just allow for detailed identification and functional analysis of the microbiome, which is what we're studying. And the purpose of the methods is to observe the microbiome's development over time and link these bacterial communities in the microbiome to health outcomes. And by bacterial communities, it's talking about the CTs we discussed in the previous slides, right? And doing this enables our ability to do a longitudinal study, or actually their ability, the researchers, to do a longitudinal study of microbiome trajectories and how it impacts health within children. All right, sample demographics. The study included fecal samples from 967 infants aged 3 weeks to 24 months living in Finland. So the purpose of this cohort is to reflect a homogeneous population to a certain area, and it mainly just looks into the population of the local geographic area, which obviously has some errors, and there are some things that could be better at this study, but we will look into how they expanded it and how they made it generalized later in this PowerPoint. And then the collection technique was gathered longitudinally, so developmental stages and health outcomes could be observed. So it was over a certain period of time, over multiple years, up until they were 5 is the effects that they were looking at. And collection took variables like birth delivery mode, antibiotic use, and infant feeding behavior into account, which could have influenced the gut microbiome. So birth delivery mode is just talking about non-vaginal compared to vaginal delivery. Antibiotic use, if the children use antibiotics over a long period of time. I'm not sure if you guys know this, but if you take antibiotics for every little infection or like bacterial infection that you guys get, like for example, if you took antibiotics every time you had strep throat, once you got something serious, it would your immune system would just, it would probably try to combat it and maybe combat it to enough of a level where you would be fine, but it wouldn't be as effective as before. And so antibiotic use when you're a child really affects your intake of antibiotics when you're an adult. And infant feeding behavior just compares like breastfeeding to a formula that children get, and it influences the gut microbiome because obviously they're ingesting it just like antibiotics. So next slide. So now we will talk about the data analysis and sequencing, to be specific. I know we talked about 16S RNA sequencing earlier. So we utilized the 16S RNA gene sequencing, or the researchers, and identified bacterial communities and metagenomic shotgun sequencing for functional analysis of the microbiome. So that's basically just saying that the two sequencing techniques that were used was 16S RNA and metagenomic shotgun sequencing, and that way they're able to gain data about the microbiome. And in terms of handling the data sequencing was processed using bioinformatics tools, which are more prominent now, and it enables people to gain data about certain things like gut health, and it ensures an accurate conclusion, annotation, and comparative analysis. And this included quality control steps to remove low quality reads and artifacts, and it basically just ensured that we had a robust amount of data integrity for more downstream analysis. Because that's important. Obviously, you want to connect this later to a generalized population of children, so the researchers analyzing stuff and producing results that could have certain implications or certain effects downstream is really important. So statistical modeling and analysis. So the model development included using group trajectory modeling to assign different pathways of the gut microbiome over time and link to specific health outcomes. This basically means that they use a certain trajectory in the microbiome and they assign different pathways to it in the gut microbiome, and from these pathways they saw health outcomes longitudinally, like we talked about. So till age 5, and these health outcomes became more and more prominent as they grew up. Like, for example, if a kid were to we'll talk about this in the results or key findings later, but if it's better for a kid to get breastfed than it is for them to drink down a formula, right? And the analytical techniques use longitudinal statistical analysis to observe the impact of microbiome composition on children's health outcome. That's just saying that it was a longitudinal study. It included multivariate regression models, and it controlled confounding variables to ensure that observed relationships were true and they're robust, and they showed up like a particular correlation, which is what the researchers were looking for. Integration of data. So how did this data integrate into our findings? The study integrated microbiome development trajectories with a longitudinal health outcome, and it revealed how childhood microbiome diversity influences health later. So just like we talked about before, they used certain trajectories in children, and they collected data longitudinally using 16S RNA and metagenomic shotgun sequencing, and it revealed how diversity influences health later, and this included synthesizing data and just having microbiome data like microbiome profiles, health records, and developmental markers that a certain gut microbiome has. And to validate, they use external data from sets and global studies. And this was a whole generalization thing that I was talking about earlier. Since it was a specific cohort of people in an area like Finland, it couldn't really be generalized, so the study couldn't be scaled properly. But like it says, the findings were validated using external data sets from global studies, and it involved a total of 5,732 infants, and it confirmed that the T1 trajectory, which just means a certain trajectory for the children, like breastfeeding or antibiotic use, could be generalized. And it basically just represented a healthy microbial development pattern across diverse population, right? The T1 trajectory was used to show and validate the generalizability of a healthy microbiome gut population in children, of course. So the overview of the findings. The study identified 4 distinct community types of CTs within the microbiota of infants, which varied with age and diet. And that's obviously how they're able to tell one implication from another. because with age and diet, a microbiome changes, and diet really depends on what you ingest, and it's different in everybody. Some people have a good diet, some people have bad. So, yeah, the study identified 4 distinct community types. So there are 4 distinct communities in the microbiomes that they looked at, and it varied. So the specific discovery is that the community type 1, and there's multiple discoveries that we'll cover later, but the specific discovery is that community type 1, CT1, was tied to infants who were breastfed and they were delivered vaginally. And it was associated with the healthiest outcome, including lower rates of, say, allergies, better immune system, and lower incidences of obesity by age 5. And then here comes the secondary findings. So community types 2, 3, and 4 were related to different environmental and dietary factors. And CT2 was linked to formula feeding, which I discussed earlier, and antibiotic use. And it showed a correlation with negative health outcomes, which I kind of implicated earlier. But yeah, health outcomes varied substantially between the different microbiome trajectories, and CT1 was associated with a healthier lifestyle factor, healthier lifestyle factors, and generally led to a better health outcome at age 5, right? And then CT2, 3, and 4 were just linked to abnormal health events like obesity, asthma, allergies, bad immune system, which really isn't good when you're a kid. So, sadly, this article did not have any figures, which made it a little complicated to visualize the results, but it's okay because I found a similar study at Dartmouth that complemented the study's results, and it kind of aligned with what they were saying. I actually found 2 studies, so we'll go over the first one first and the second one second, obviously. So yeah, this figure from Dartmouth shows a correlation between infant microbiome diversity and early childhood behavioral outcomes, and it's differentiated by sex. So if you look closely into the study, let me look in here. You can see, for example, like anxiety and depression. For women, it did get better as time went on and as they grew. But not as substantially as men. For men, it got a lot better with a good gut microbiome. And the whole visual interpretation is just that this graph could represent how increased microbiome diversity correlates with certain characteristics in children, like the anxiety and depression that I talked about. And it just highlights the whole complex interaction between the microbiome diversity and child development. Next slide. And this is the second figure that kind of complements this study because my study didn't have any figures. But the figure portrays the interactions between the microbiota and the developing brain. It focuses, if you look at the figure closely, it focuses on the critical window from birth to 3 years, and this study focuses from birth to 5 years. So it kind of is a little bit more extensive, but it basically just shows how microbial colonization and early life exposures contribute to brain development and immune training. So it kind of fits in with the whole immune system thing that we were talking about, and the whole visual interpretation is that it demonstrates how the gut microbiome influences neurodevelopmental outcomes, and it just demonstrates the complex pathways that the gut microbiome goes through and how it influences neurology in your brain, and it highlights the role of microbial metabolites and immune-mediated mechanisms in shaping the whole neuroimmunity framework and the brain developing from 0 to 5 years. 0 to 3 years in this study. But it just shows that the microbiome plays a big factor in all of that, and it's really prominent in that age range of 0 to 3 years. So, in our discussion, we're going to be looking at the synthesis of the results, and this study just gives evidence that the composition of the gut microbiome in its early life is predictive of long-term health outcomes. It highlights or underscores the importance of microbial diversity and a stable microbial community in influencing health trajectories, particularly in preventing asthma, allergies, obesity, and the immune system, stuff that we already looked at. And the whole outlook or future outlook is for this research to introduce a treatment for targeted interventions, so the whole preventative healthcare aspect, and we want that to manipulate the early microbial activities and improve health outcomes. Future studies might explore more specific mechanisms in which microbiome diversity can affect healthcare, and doing that would just validate everything globally. They could look into like breastfeeding, for example, specifically look into antibiotic use, and stuff like that. So next slide. And then the whole broader implications is in regards to policy implementation. The study's findings could play a heavy role in public education regarding public health strategies and just foster a communal support of early microbial intervention. And doing this could just encourage a healthy gut microbiome in infants and promote breastfeeding and not the eradication of antibiotics, but just using them carefully. So the directions for future research would be exploring the different pathways by which the microbiome diversity impacts long-term health outcomes. And I talked about this a little bit earlier, but it could investigate the impact of different feeding practices and the environments that the children grew up in. So regarding the whole conclusion of the study, I'll talk a little bit about what the slide says, and I'll expand upon that to tie everything together. So in terms of what the study contributes, the study grows our understanding of how early life gut microbiome composition influences long-term health outcomes. And basically what that's trying to say is that by identifying specific microbiome development trajectories and patterns and linking them to child health outcome, it provides us with the foundation for future interventions in the whole preventative healthcare aspect. And it just overall aims at enhancing pediatric healthcare and bring a new type of possible future to it, one that is more considerate of a pediatric patient's long-term health outcomes. And then in terms of the healthcare impact, the findings suggest that early microbiome interventions could provide us with a new strategy in pediatric healthcare, thus preventing a multitude of chronic conditions. And I discussed this earlier, but these new microbiome interventions and new strategies in pediatric healthcare gives us a whole lead on a paradigm shift in how pediatric healthcare is approached and emphasizes prevention over treatment, and just highlights the role of what the gut microbiome does from an early age. And next slide. So these are just some thoughtful questions for discussion. So question 1 asks, what are the challenges of monitoring microbiome data in regular pediatric checkups? And to answer that question, challenges in microbiome monitoring include just like the complexity and the whole cost aspect of microbiome sequencing, which limits its use in routine pediatric care, the lack of standardized protocols for analyzing and interpreting data complicates its integration into clinical practice, and additionally, determining actionable steps from microbiome data is complex, and it demands a clear understanding of its clinical relevance, which is still evolving, still in the making. And then the question that asks about how can this research be communicated to a multitude of audiences, I would say that it can be effectively achieved by relaying it to a large number of people by using simple language and visual aids. Infographics, just advertising, the gut microbiome research and the role gut microbiome plays in pediatric patients really helps illustrate the impact of, or portray the impact of gut microbiome on health. And we can hold workshops, seminars, and just provide interactive opportunities, and both for patients and educators to learn from experts. And it ensures that the information is both accessible and engaging. And then, in terms of ethical considerations and what ethical considerations come into play when predicting health outcomes based on early life microbiome data, we just need to kind of follow the whole HIPAA thing. We need to include ensuring the privacy and protection of sensitive health data, obtaining consent from parents, because we're talking about children here, and just addressing the potential for certain determinary interpretations of the data, which could lead to a certain stigma or anxiety that the parent has, or maybe even the child if they tell the parent about a child's future health. And then in terms of factors that should be considered when developing interventions based on microbiome data, which is what the fourth question asks, what factors should be considered when developing interventions based on microbiome data. We should include the variability in microbiome composition in the individual, because obviously everybody is built different. The influence of diet, like I said, and lifestyle, or the environment that they grew up in, has a big impact on the microbiome, and that's why we need more research, like I discussed, to look into the long-term effects of these interventions and just ensure that they're beneficial and safe. Last question: how might this research impact new dietary guidelines for children? The impact of this research and other research that falls along the area could just lead to a personalized nutrition plan, depending on the microbiome data that they have, the diversity, and how their gut health looks. And that can just tailor dietary recommendations to individuals regarding their microbiome profile and enhance the child's health from a very early age, so they can develop healthier and grow really healthy once they become an adult, or even by 5 years, which is what the study was looking into. And this research could also influence public health policies by integrating tools, and these tools would just support a healthy microbiome. And an example would be like probiotics, foods that have high fiber, prebiotics, and just other dietary guidelines like that fall into that range where gut health is promoted and wanted from parents to their own children, so they can live a healthy lifestyle and have a healthy future. And just doing all of this as a whole, this research would just impact pediatric patients and children into having a good lifestyle in their future and improve the whole preventative aspect of healthcare in children, which is what we want. We don't want to react to their illnesses. We want to prevent it when a sign of it comes up. And the next slide is just the references. These are all the references I used in the article, excluding the article itself, which was listed on the title page. Thank you. I like this presentation a lot. I loved making it. I spent a good amount of time on making it. Thank you guys. I hope you liked it.